This is Dr. Mubin Sayed with one more episode of Long Story Short with DrBeen from the FLCCC platform. So the discussion today is about proteins, heat shock proteins, autophagy, controllers of autophagy, chaperones, and the chaperonopathies. What is this all? So we have done in the context of autophagy, intermittent fasting, and removal of intracellular trash. In that context, we have done a few talks before as well. And I felt, Dr. Paul Merrick and I was discussing about the autophagy. And as we were talking about various mechanisms, I thought it will be interesting for medical students, nursing students, healthcare providers, and general public to understand and connect these mechanisms together. So when they are thinking about management of a patient, they can actually connect the dots and understand what levers to manipulate and what to expect. And of course, if you are thinking about long COVID or vaccine injury, and I would suggest that even chronic inflammatory diseases, then the FLCCC.net is a great place to see various protocols there where Dr. Paul Merrick and team has done all of this exercise of looking at the mechanisms and then vetting out those therapies that are promising and then putting them together and having them available for you. So I would strongly suggest that you look that up. Let's start our discussion. So again, the purpose of the discussion is to understand in the context of autophagy, what are the proteins, heat shock proteins, and what are the mechanisms behind autophagy. So proteins, heat shock proteins, and autophagy. What we'll do today is we'll talk about proteostasis. Proteostasis is when we have proteins that are being made in our cells, then these proteins function, then those proteins are broken down. This production of proteins and breaking down of protein is always in balance. We do not produce more protein than we break down. That would cause overgrowth of proteins. Similarly, we do not break more than we produce because that would cause tissues to start getting damaged. So there is always a balance between them, and that balance is called proteostasis. Then we'll talk about chaperones or heat shock proteins. We'll talk about the stressors on the cell that modulate the presence and function of heat shock proteins or chaperones. Then we'll talk about protein recycling mechanisms and the type of mechanism and ubiquitination versus lytic or autophagy mechanisms. We'll look at some of the controls for autophagy. And finally, we'll look at some of the chaperonopathies or issues that can occur with the chaperones that in turn would cause cellular damage. So let's start. Lots of things to discuss. So significance of this presentation, why are we talking this all? The point was to thread together the foundations all the way to autophagy. So let's start from here. In our cells, when the cells are functioning, these cells make the machinery to do a specific function or a specific role that needs to be performed within the cell or outside of the cells. So for that role, in that point in time, the cell needs to make proteins or enzymes or machinery that would take care of that function. And then that machinery, once produced, will do the function. And finally, that machinery will be torn down. Our cells are very strange compared to us. For example, if a cell is going to cook something, it will have to build the pot in which to cook something. And then it will cook. And then it will eat whatever is cooked. And then it would destroy the pot. And if it has to cook again, it will build the pot again. So cells make their machineries again and again. So if you see here in this diagram on the left side, this is a ribosome that is present inside a cell. And the ribosome, this little red thread, is the messenger RNA that is attached with the ribosome. And the black thread that is coming out of the ribosome is the nascent protein being built. So this is a newly forming protein. It is not yet even formed fully. It is just coming out of the ribosome. And as the whole messenger RNA is red, messenger RNA is the recipe. As it is red, then that protein formation will become complete. So once that protein thread is completed, that is called a nascent thread. It's nothing but a thread, a cord, a string. Then what we need to do is we need to use that string, that yarn, if you will, and shape it into some structure, three-dimensional structure that can do some function. So if you see here on the right side, from this yarn or string, we have created a three-dimensional structure, which is a native protein, and that has a function to do. So if you go back to the thread, so on the left side here, the protein is in the thread form. Then what happens is it becomes either a coiled form or a two-dimensional paper-like structure. We call it beta-pleated sheets. And then from these secondary structures, the tertiary structure is formed, which is the three-dimensional structure. And sometimes these three-dimensionally shaped proteins, they attach with each other, multiple of them, and they make even suprastructures. This is like you bring in the beams and cement and other things and build a house with them. So here on the right side, you see quaternary structure, where multiple three-dimensional proteins have come together and they're making a channel in this situation. So it is important for this thread, that black little string, to become correctly shaped into a three-dimensional structure. That is called folding. So imagine in your mind, as we are talking about these discussions, as we are talking about the proteins, imagine a person sitting with a ball of yarn and is knitting a sweater. Yarn is a single-dimensional string, and the sweater is going to have multiple sheets. Then those sheets will be connected and stitched together to make a three-dimensional shape. So here, if you see on the left side, I have added another bowl over here. This bowl is a chaperone protein. So as the ribosome makes that little string of protein, that string is not just running around in the cytoplasm, freely floating around, but instead it is captured in a chaperone. So there is a chaperone attached to the ribosome, and as the string comes out of it, the chaperone keeps collecting it. Once it collects the chaperone, collects the newly formed protein, then that chaperone uses energy to shape this string into a three-dimensional protein. So that is what you can see in the middle diagram, that string has now been converted into a three-dimensional structure. Then that chaperone, if you see the right side of the diagram, the chaperone is now dissociated from this 3D structure. Some chaperones do not dissociate. They actually carry the protein to its destination as well. So they act as a vehicle as well. But here I've made the chaperone that is dissociated. So here is a little cartoon for you that there is a chaperone protein, and that is saying to this newly built diaper wearing small baby protein, it is saying, "Darling, your biological mom is a ribosome. I carried you in my womb." So here, sometimes when the protein is being built, even during the time of building, it could become misshaped. And this misshaped protein is given an opportunity to still become correctly shaped. And how is that opportunity provided? If you see in the center, this little multicolored structure that I've made, that is called a chaperonin. This newly built misfolded protein is inserted in a chaperonin. You can think of that as an incubator for a premature baby. And that chaperonin has environment inside that allows the protein to uncoil and recoil, or unfold and refold. So there are enzymes and pH levels and temperatures correct enough to allow this misfolded protein to unfold and then refold. And it is possible that after this living in the chaperonin, this special care in the chaperonin, the protein would then correctly get folded into a three-dimensional shape. But here I want to plant a seed in your mind, and that is, it is possible that the protein got misshaped, and that misshaping was so bad that it could not attach to a chaperonin, or it did go into a chaperonin, but even after being there, it stayed misshaped or became even more misshaped. So keep that in mind, because we'll have to do something about that protein that is now damaged or misshaped. So now this newly built brand new protein, which is now properly folded, it has a 3D structure. That protein now needs to go from the area of birth. This all is happening within the cell. From the area where it is born to the area where it will perform its function, and then it will be recycled and destroyed. So there are more heat shock proteins whose job is to pick up the newly built proteins and take them to their destination where these will perform their function. So imagine if there is a shop or there is a factory where there are some things being built. So there will be a pile of those things over there. And then you need vehicles and other mechanisms to pick up those things and bring them to various shops and various places where they would do their function or be sold, eventually to go somewhere else to perform their function. So within the cell, bringing a newly born protein to its destination within the cell to perform its function is through the chaperones. Sometimes the chaperones will bring this newly born protein to another organelle, another factory, where this protein will be further packaged and sent outside of the cell as well. So do you see that in this whole process, there is a specific mechanism that is important, and that is the chaperones. And we are talking about chaperones as well. So now here, if you see in this diagram, imagine this protein has been brought to its final destination. And now it is working there. So in the center, do you see that there is this little protein standing there with its worker hat on, and it is doing some work. And there's a little cone here that says work area. But if you see, there are some monsters that are attacking it. It is possible that during the functioning, a protein might get damaged while performing its function. Or a protein might get damaged because the cell became toxic. How? For example, let's say a virus arrived in the cell. And now virus is producing proteins that are going to attach and attack other proteins. Or the environment became acidotic. Or for example, reactive oxygen species increased, or reactive nitrogen species increased, and those species are attacking the proteins present in the cell. And now this protein, which is actually healthy protein, it was built correctly, it was functioning, all of a sudden this protein is going to become damaged because it is now attacked by whatever are the circumstances, toxic circumstances. So here, if you think about it, for a protein, from birth to function to destruction, so from cradle to grave, the service to this protein for the maintenance of the protein is provided by chaperones. I've done this talk once before as well. I'm just taking this little piece from that talk. So this should tell us that protein folding, bringing them to the correct place, having them function correctly is very important. And the chaperones are very important. And if chaperones themselves are sick, then imagine what havoc will occur within the cell. So now misfortune of misfolding. It is possible that a protein, as I said before, is produced badly and it is misfolded, and even chaperonin cannot help it, or the chaperones cannot help it. All the mechanisms that help shape it have failed. Or, as I said, protein was functioning and it got damaged because of a reactive oxygen species and other toxic substances. Or it is possible that protein has done its function and now it has reached a point where it has become nascent and it has started unraveling, it has started uncoiling, its shape has started becoming bad, it is getting out of shape now. It is also possible that protein is actually fine, but the chaperones that are helping the proteins maintain their shape, those chaperones are overwhelmed. For example, imagine a cell is living in a toxic environment, or there is a virus that has arrived, or there is some stressor outside of the cell, or there is some acids, or there is less carbon dioxide, oxygen, or more carbon dioxide. Many kind of stressors are possible. Or the cell is under control of hormones, and hormones are overwhelming the cell to produce more proteins and so on. In this case, it is possible that the chaperones that are serving the proteins, these chaperones are overwhelmed. There are so many proteins produced, or there is so much damage done that there are, let's say, 1 million protein molecules that are damaged, and there are only 10,000 chaperones which need to fix them all. Now those chaperones are overwhelmed and the misshaped proteins are lying around. So these lying around proteins that are misshaped, that is a problem for the cell. Cell will have to quickly correct them and use them. Otherwise, these will need to be recycled or destroyed. And if they do not get destroyed quickly, they will become tangles. They would start tangling with each other. And now that trash, have you ever seen sometimes within our homes, there are the cables, and there are like 20 cables all tangled with each other? That is what is going to happen. So this is a very delicate point now. A protein that has become misfolded, how will we recycle that? So first we'll try to fix it. If we cannot fix it, we need to now recycle it. How will we recycle it? So please keep in mind there are two mechanisms. One mechanism is called ubiquitination and the other mechanism is called lytic or autophagic mechanism. We are going to look at these two mechanisms now. So here, before we go to those mechanisms, the pathway to them. Imagine this side here is all the chaperones of the cell. The chaperones present in a cell are collectively called a chaperone hub. They all collectively make a platform of social services within the cell. Chaperone hub. That chaperone hub is functioning normally at a baseline, but when a cell is producing more proteins or is under stress and the more proteins are getting damaged, or when the cell is functioning at a hyper state, for example, it's an immune cell and it is fighting with a virus or other cells that are infected or cancerous, then the cell is functioning at a hyper speed. In all of these cases, as I discussed before, proteins can be misfolded, chaperones can be overwhelmed, chaperones themselves could be not correctly working. So now what happens is there are two pathways. If a born pure protein by itself has become misfolded, then we can tag it to be destroyed, and that mechanism will be ubiquitination. So if you didn't get it yet, let me explain the other part and then this will become clear. On the other hand, so when multiple proteins have become misshaped and chaperones are trying to shape them, but the chaperones themselves get trapped in those misshaped tangles. And now we have a pile of garbage where there are multiple proteins, not a single protein, multiple proteins with multiple chaperones all tangled together. Then this mess cannot be handled by ubiquitin pathway. This will have to be handled by autophagic pathway. So if there is a single protein that is misfolded, ubiquitination can be done. So that is a normal physiological function. Even autophagy is physiological as well. However, autophagy is more involved, more sophisticated because it takes care of the trash or tangles that are formed. So if you see here, the pathway above is one protein and it is damaged, and so you can see that it is marked. These little red things are markers on it, and these markers are the ubiquitination markers. This protein has been marked to be killed or to be destroyed, recycled. The pathway below is a mess. So there are many three-dimensional proteins with many fibers and chaperones all stuck together. This mess, this pile, this garbage will have to be piecemeal removed and destroyed. So for that, if you see there is this little donkey, there are special chaperones that will pick up pieces of this garbage, bring them to special lysosomes or digestive systems where these pieces will be recycled. This is equal to trash removal from a house, and then the truck brings that trash all the way to the trash processing unit where the trash will be recycled. So here, ubiquitination. So if you see here, these purple proteins that are all trembling and are a little nervous, these proteins have been marked to be recycled. To recycle them, the intracellular organelle is a proteasome. A proteasome is where these proteins are brought. And this is a process of ubiquitination. Proteasome will then disassemble these proteins down to their amino acids, the individual bricks, and then those amino acids will be used by to build other proteins. So this is ubiquitination. On the other hand, if we have to perform autophagy, that is, for example, let's say in a patient there is a spike protein stuck or S1 protein, or the virus is present and there are so much viral proteins that are present. In the beginning, the cell would actually put up defenses and try to clear out the viral proteins. Eventually, the virus can overwhelm the chaperones and the folding system and the autophagy system. And in case of SARS-CoV-2, for example, the spike protein and the other proteins in the virus can actually turn off some of these mechanisms that are helping to clear out the virus. And that way, virus will not be removed as a trash. So here, if you see, there are some proteins, for example, ATG7, autophagy protein number 7, autophagy protein number 5, and so on. And then you see some phospholipids. So these proteins are present with the cell membrane. The point is that when autophagy is needed, then some of the autophagy proteins, they build the phagosomes. So again, sticking with the analogy of trash removal, imagine there are workers that will build the trash truck, the phagosome, the structure in which the trash will go. Then that structure will be brought to wherever the recycling is needed, will be done. So here ATG7 is building a phagosome. Then eventually, and if you see here, the beclin is standing here as well. Beclin is also a very important controller of autophagy. So if we see here, this is a more involved picture, but as you can see, there are multiple controllers for autophagy. mTOR system is one. Then if you see here, this is beclin, this is another. Then AMPK with ULK. These, all the complexes work together in a great balance to decide when the autophagy would start. And when they decide that autophagy would start, then if you see here, a small piece of wall is made, then that wall becomes bigger to become a bigger whole vesicle. This is a phagophore that was forming. It will become a phagosome, or called autophagosome, which would then be connected with the lysosome or digestive system, and that would be called autophagolysosome, and that is how the trash is removed. It's such a sophisticated mechanism that these little trash packaging bags or phagosomes are constructed near the trash. So imagine there is a patch of trash and we bring materials to develop a bag near that. So we go near the trash and we develop a bag, and when the bag is being developed, it engulfs a part of the trash, and that is a piecemeal removal. And then it goes to the lysosomes and gets destroyed. Beautiful mechanism. And if you see this diagram, there are various, for example, resveratrol or spermidine or coffee. All of those are actually controllers or modulators of beclin and mTOR pathways to eventually trigger, allow the autophagy to occur. Of course, intermittent fasting. Of course, stresses on the cell. A cell that is under stress with heat or temperature differences, or with physical or mechanical stresses, these pathways will become triggered and they would allow the autophagy to start occurring. Now, you could say that when the cell is under stress, it's not necessary that autophagy is needed, but the cell would start preparing for autophagy right away because it knows that stress is here, more proteins will be produced, some proteins will be damaged as well. So I will have to have the social workers present to clean out. And then we have done this discussion before as well. I've borrowed those pictures here. The type of autophagy. The autophagy is macro, micro, and chaperone mediated. And I hope that we now have a full picture of how a protein is made, how it functions, and then it might end up in a trash. And now we need to remove it through autophagy. That is the only way to remove it more successfully. And the ubiquitination will not work. So here, if you see, macro autophagy, the diagram on the left, is where a phagosome is formed. The trash is removed using autolysosome. On the right is a very interesting thing. There is a little Pac-Man like lysosome, the stomach, that is nibbling at a cell's organelle. This I have made Golgi apparatus, for example. So there are other organelles as well, ribosomes and endoplasmic reticulum, smooth and rough and so on. So what happens is when a cell is functioning, it may need to process a lot of proteins, so its organelle might increase in size to accommodate extra function, extra production needs. Once that need is gone, we may have to trim down these organelle, otherwise they'll just keep becoming bigger and bigger. So we can do microautophagy that would trim these organelle. This is like a gardener that trims the plants to keep them in shape. So here we are keeping in shape the organelle, cell's own structures, instead of trash. So that is microautophagy. And finally, chaperone-mediated autophagy, where the trash is picked up and brought to the lysosome, as I mentioned before. So before this last diagram, I hope this becomes clear that the role of autophagy is needed to remove the more unremovable trash pieces that are formed from damaged proteins which are damaged either by birth or damaged during their function because of, let's say, reactive oxygen species or viruses or the virus proteins that have turned off the recycling mechanisms. And so now we have trash in the cell. And if we can trigger autophagy, then that trash can be removed and the cell can be brought towards a normal state, which would reduce inflammatory state of the body. So chronic inflammatory diseases can benefit from these mechanisms. So finally, one more slide, and that is chaperonopathies or abnormal chaperones. So it is possible that in some people, the chaperones are also proteins and they also have genes, and those genes give rise to eventually these proteins through the messenger RNA and ribosome. Sometimes those genes are damaged. So the chaperones that are produced are not correct chaperone, and that can cause many disorders, genetic disorders, neuropathies, cardiac diseases, liver, kidney diseases, and that is because chaperones were bad. Or it is possible that somebody was born with the correct genes for chaperones, the chaperones were working correctly, but the cell is under stress, or there is acute infection, or there is spike protein stuck somewhere, or there is some other toxic substances, and the result is that this healthy cell's chaperones have become bad. For example, impaired stress response will create overwhelming demand for chaperone. So imagine if the spike protein comes into our cells and turns off the stress responses. That means now the stress responses are not taking care of the spike protein and not doing recycling of it. So there'll be piles of trash. And now the chaperones are overwhelmed. Then similarly, high number of misfolded proteins. Let's say reactive oxygen species are damaging a lot of proteins inside the cell. Chaperones cannot take care of all of them. Imagine incorrect pH. So normally enzymes work at more acidic pH within the system. And now if we incorrect the pH, or the pH becomes more basic, or pH becomes more acidic than it should be, then the chaperones will not work correctly. Similarly, pathological increase in the reactive oxygen species volume, then lack of substrates for nitric oxide, which helps with reducing the reactive oxygen species, and sequestered proteins, toxins, aging, cancer, cardiac diseases, neurological diseases, and other can occur when chaperonopathies occur. It is actually said that if we can take care of autophagy and if we can have healthy chaperones, then our aging process reduces as well. It slows down too. We become healthier. And you can actually rejuvenate the cells by clearing the trash out of them. So there are lots of other topics that I've already done about autophagy. I wanted to connect these dots here. Please watch other videos too. And with this, thank you very much. I will see you with one more episode the next time. So bye for now.